in a healthy person, because somatic mutations that accumulate during a lifetime are often associated with cancer. But how many mutations do we accumulate that do not associate with cancer? What is the background somatic mutation rate that we can endure and still be healthy? So what we did is we took a very old person, to have a lot of time to accumulate mutations, and we had access to her peripheral blood, so we did not have to culture the blood, and we went to see if there are somatic mutations in this peripheral healthy blood. And I'd like to introduce the person that we used for this. This is Mrs. Hendrikje van Andel-Schipper. She was a supercentenarian. She was 115 years old. Okay. And this is a little bit of an introduction. So she was born in 1890. She was born very prematurely at 1600 grams. She did not have any children. And at the age of 82, she decided to donate her body to science. And at the age of 100, she had a breast tumor, which was removed. If that wouldn't have happened, of course, she would not have turned 115 at the end. But when she was 112, she had remembered that she had donated her body to science, and so she called the hospital and said, "Am I still valuable for science?" So people went to visit her, and they asked her all kinds of questions, and they wanted to note how well her brain still functioned at 112. And then they went back at 113, and they saw that her brain was still functioning very well. So this is a very healthy person. I mean, there was not much brain degeneration. And at the end, they actually could show that when she donated her body to science. So she died eventually at 115 of a metastasized gastric tumor. She had no heart disease, no Alzheimer's disease, and a healthy blood compartment. So what we did is we wanted to take a fast-dividing tissue, such as blood, and compare it to a slow-dividing tissue, such as brain, because we think that the most mutations occur during cellular division, so most mutations would have probably taken place in the blood. Whereas brain cells hardly divide, so not many divisions, maybe not so many mutations. So that's the two tissues that we decided to compare. So to see if our assumption was correct, we decided to check the telomere length of all different types of bodily organs, and we saw indeed that the brain had very long telomeres and that the blood had the shortest telomeres. So perhaps the brain had much fewer divisions compared to the blood, and that other organs had divisions times in between. So how many mutations actually occur in blood? So what we did is we took the blood DNA and the brain DNA and we sequenced the entire genome. We did this 60 times for the blood and 60 times for the brain to get enough statistical power to get good mutation calling. So the mean read depth for this was 60 times, but there, of course, there are regions in the genome that are sequenced less efficiently. It took us about 4 months to get all the sequencing done, and it took about 10 terabytes of data storage plus compute, so it even blows it up even more. So this was a pretty big exercise to do this on only one person. So indeed, we defined the mutations that were in the blood but not in the brain, or in the brain but not in the blood. And we found single nucleotide variants, 612 of them in the brain, and that were not in the blood. And in those we found 23. And vice versa, we found 107 mutations in the brain that were not in the blood, and 3 indels. However, there are mutations that were in subsets that were not as deeply sequenced as 60x or maybe more. So we have to validate for each of these if they are actually true mutations. So then we found that indeed in the blood there were 424 mutations that we could validate with a different method and 22 indels, but in the brain there were none. So we could say that in her lifetime, in her 415-year lifetime, she had accumulated about 450 mutations in her non-repetitive genome, because that is what we assessed. If you extrapolate that to the complete genome, that would be about 600 mutations in our whole genome. Except, we think this is very much a lower bound estimate, because the repetitive genome is much more mutation prone than the non-repetitive genome. So this is very much a lower bound estimate. So where in the genome do these mutations occur? So what we did is we did an enrichment analysis using the ENCODE data. That's a database with all kinds of functional DNA elements, regulatory elements. And we wanted to see if where in these regions the mutations occurred more or less. So there was a gene enrichment in gene-poor heterochromatin in the lamina-associated domains. So that's depicted here. There are parts of the genome that attaches to the lamina of the cell, the nucleus, to keep the chromosomes in place. And in these regions, there was a significant increase in mutations. There were also a significant increase in mutations in the bases that were accessible for solvents. So they were sensitive to ionizing radiation. And they were at methylated cytosines, at CpG sites, for spontaneous deamination. So that's actually all to be expected, because these are regions that don't really matter that much if they are mutated. And they're also depleted in regions which may have adverse effects on cellular fitness, such as all kinds of regulatory regions that open up cells for transcriptions, the DNA for transcriptions, such as activating histone modifications. Then we also checked to see, there's of course a huge dbSNP database on SNPs that are recurrently present in all of our genomes that are completely harmless. So where do they occur? And we did exactly the same analysis with dbSNPs and also for mutations that were shown to cause disease. And we saw that the mutations that we found that accumulated during her life occurred at the same loci as the mutations that are in all of our SNPs, so that occur in the germ cells. And there are different loci compared to ones that do cause disease, because they are much more in activating or gene or in regions where genes are transcribed. We found 4 mutations that occurred in the coding regions, in the coding genome, and there was 3 missense mutations and 1 nonsense mutation. And these were the genes that they occurred in, but we couldn't really associate that with anything that could be harmful or maybe protective or anything. They didn't show any reason for us to think that they were important for this to happen. So then we decided to look, how are these mutations distributed over the genome? Or actually, what's the variant allele frequency of them? So when you have a normal germline heterozygous variant, which we all have, you get one from your mother, you get one from your father, that means that there is a variant allele frequency of 0.5. But when you measure that with sequencing, you don't usually get a line at 0.5 unless you go very, very deeply. So you always get a distribution around 0.5. So this distribution is for cells, for variants that are heterozygous in 100% of your cells. And the mutations that we found that were verified had this variant allele frequency distribution. So that means that they are not present in all of the cells, but they are present in a pretty high fraction of the peripheral blood cells. So when we started to fit curves on this distribution, we saw that there were actually 2 curves. One at around 0.22, the variant allele frequency, and in the second one, the mean variant allele frequency was at 0.32. So if you think about that, that means that this first peak, the A peak, is heterozygous in 44% of the blood compartment. 0.5 again was when there's 100% heterozygosity, and 0.22 would be 44%. Same goes for the second peak, 32, means that in about 64% of the blood compartment there are heterozygous mutations. This, of course, does assume that we are looking at heterozygous mutations that she acquired during her lifetime. But the problem there is, if you would add those 2 percentages, 44 plus 64 is 108%. That is not possible. So maybe is the 1 one clone a derivative of the other clone? Is there an older clone and a newer clone? Or maybe they are 100% together, there are two clones. We figured, well, maybe we're looking at a clone and a subclone. So we think that perhaps B is the older clone because all the mutations occur in all of the cells, and A is the larger clone, but a subclone from B. And that together we were able to pick up about 64% of the peripheral blood genome, or the mutations in the peripheral blood. And that 36% was not being able to pick up. So what does that mean? Estimates say that we as humans are born with about 20,000 hematopoietic stem cells that remake our blood, and that about 1,300 of those are simultaneously active during a lifetime. So two active hematopoietic stem cells is very small. It's very much a decrease during her life. So then we ask, does the number of hematopoietic stem cells that are active decrease during aging? So for us that was a good thing, because otherwise we would have never been able to detect the mutations in the first place. So the fact that she only had two active stem cells allowed us to detect the mutations. But now we want to know which type of blood cells are left. Which type of blood cells are regenerated by those two active stem cells? Unfortunately, we were not able to check this, but these of course are the questions that arise from these results. So when you think about that, there's two types of, well, there's many types of blood cells, of course, but there's the lymphoid system and the myeloid system. And the myeloid system is short-lived. There are cells that are alive maybe 6 days or 10 days or whatever. And then there's long-lived cells, and they can be alive for maybe 60 years. So perhaps what we're looking at is that perhaps the fraction of myeloid cells increases over a lifetime, and the fraction of lymphoid cells decreases over a lifetime, such that what we picked up is only the short-lived cells that are necessary to keep us alive, to transport blood and to transport oxygen or whatever, and that the T cells and the B cells are older, and that that are remaining in the blood that we couldn't pick up because they were probably made up from stem cells that were active much earlier in her life. So in our future work, we are going to try to answer these questions. So we want to know, are the short-lived myeloid cells the types of cells that were actively generated by the two active stem cells? And are the ones that we couldn't pick up, or were they perhaps generated by active stem cells that were active much longer ago and not active anymore? Maybe they're inactive now, or maybe they died. Let me see. Right, so that's the same thing. So that's actually what we're doing right now in a new study. And this time, so we also took the possibilities to cell sort before we sequence the genome. And we are currently looking into this, so hopefully we'll have data for that soon.
And thank you for running quick.
Yeah.
We're almost back on schedule, so we have time for questions. Sir.
How can you exclude? Sorry. How can you exclude the possibility that the two cells, or the clone and subclone, were actually in cells that were very early on in embryonic development, before the creation of the hematopoietic system? In other words, that they there may actually have been a lot more than two actually active long-term repopulating cells in the lady, right. But they came from
they were already cloned.
Yeah, exactly. So you're saying they could have been there, present we don't know. We think that they must have accumulated during a lifetime, but we cannot exclude that they were already there
really early.
Really early, of
right,
course,
so,
yeah.
Yeah, because then, I mean, you could have had a perfectly normal number of active stem cells.
Well, but then still there would be two different types of clones.
Yeah.
That would still be the case.
Yeah, that's what you got, yeah.
Yeah, so there would be definitely two clones, or one stop clone or whatever, that could be the case. We can never be sure when those mutations arise. Everybody always asks, so what is the mutation rate? Well, I can take that number and divide it by 115, but I don't know when they occurred. I mean, I have absolutely no idea.
So how are those numbers, like 20,000 at birth and 1,500 later active, how are those numbers derived?
That were from all kinds of models that were run from mouse studies, and actually I think they were also extrapolated to human situations. I think these are all estimates that are, I don't know, if yeah, so those are all estimates. I think I should keep it at that, yeah.
Hi. So I have two questions actually. So, thank you. Thank you. Very interesting. So you mentioned the mutation rate, and there's been other studies using next-generation sequencing to estimate mutation rates in individuals, none as old as the one you have. The question is, how does your the number of mutations you're finding compares to that? Because I believe there were 2 Nature genetic studies, for instance, that suggested that it's quite not a lot of mutations accumulate with age.
Well, so we've done that comparison. I show it up here. So when we divide the amount of mutations with 115 years, we get about 4 or 3 in a non-repetitive genome, depending on per year or per division. However, with healthy individuals, from the studies that you're mentioning, they come up to about 9 mutations per year. So we are on the lower end to that. We don't know if these are. I think it's really difficult to compare because of the way that we sequence, the way that we do the measurements are all different between all these studies. So I think it's really difficult to compare these numbers. But we're in the same range. So I don't know if she's extremely healthy, having less mutations per year or per division than others. But I cannot say that that is because of a technical difference or because of her extreme health.
But how do you calculate? You divide it by 115, right?
That's what I've done here. I mean, like I said, I have no idea to the rate at which these mutations occur, if they're lower during childhood or later.
Yes.
So I have no idea how to check
There's
that.
a Nature Genetics paper where they do that for the different individuals of different ages, and what they suggest is that it's quite low, the mutation rate per year. Well, they show the mutation load at different ages, and it's pretty much stable until you reach about 50, 60, and then it increases slightly.
So then that would still, this is a correct assumption,
Yeah.
then if that would be, true here as well.
Yes.
Yeah.
I have another question, but if someone want to take over all the questions.
Got a couple of minutes.
Okay, so my other question is, you sequence the genome of this individual, very long-lived. So my guess is you could probably check if there's particular genetic variants in this individual that could be related to longevity. Just check, known longevity-associated genes, whether they have any variants in them.
Yes.
Done that.
We have done that, except we didn't find anything that was, different from the normal population. So yeah, there was one variant in BRCA2, but we don't know if that's a protective variant or not. That's actually also in the paper. So if you would like to see all those genes that we did find, it's in there.
Okay.
Yeah.
If I understood correctly, you didn't find any mutation in the brain. Is this an artifact of your technique? Because if there are mutations, there are no. The
Right.
size of the clone is so limited that you don't pick it up, or
I think that so we took a small part of the brain, and that consists of many different cells. So if there would be mutations in there, probably maybe if it's present in 1 or 2 cells, we would never be able to pick it up with 60x sequencing. So probably they are there, but it's not a clonal situation, so that therefore we couldn't pick it up.
so it's just an artifact. It's something that you expect.
Well, okay, maybe I should have rephrased saying we did not find any clonal mutations in the brain genome. Yeah, you're correct. Yeah.
One more question. You said that she was born prematurely.
Yes.
Do you actually know she was born prematurely, or only that she was born very small?
No, actually she was born very prematurely, and actually her mother thought that she would die, and so her grandmother had her in her skirt and gave her cow milk, and that's how she survived. Yeah.
So did she struggle during her reproductive years a lot with infections? Is that known?
No, I unfortunately did not know her. I don't know if that is the case. The fact that she didn't have children has nothing to do with the fact that there was a bodily problem
No,
or
but that's
anything.
not why I'm asking. It's just, did she have maybe was she maybe weak in her young adult?
In her young adult, she was kept home from school because every time that she went to school, she was ill. So she was kept
That's
home.
the thing.
Yeah. But at her later years, she was just perfectly fine.
Yeah, so that's related to this theory about successful aging, that if you have, for instance, a very good immune system at a younger age, to get you safe through the reproductive period, etc., that you have actually a lot of inflammation at later time. But if you have a lot of infections during your early adulthood, you could say, and you get through that,
Yeah.
then you probably have a better, well-balanced immune system. That is one of the theories that's put forward. So I was wondering.
Could be
in
the
relation to
case
that.
here. Yeah. Could well be. Yeah.
Nice example. Yeah.
I don't think it has been asked, just to let me know if, could it be that her two cells are in fact the explanation for her longevity? Could it be that there has been a sort of selection as when we do immortalized cells, because she was maybe a bit fragile in terms of immune system? And I don't know, I mean, do you have elements indicating that her case is really special or not yet?
So I'm not. I don't think anybody else has really looked at these extreme ages of how the immune system there works. But so the only thing that we could do is, with the two cells that we do have, look at where the mutations are, and if they could give any idea for us to think that may be protective or that may elongate her life. We did not find that. But perhaps it's in there, but we haven't found it as of yet.
I think we're going to have to move on to our next speaker now.